It's really my pleasure to host Kazuhiro Maeshima. He's currently the head of the Department of Chromosome Science at the National Institute of Genetics in Japan. And he's had a long and productive career working on gene regulation, cellular dynamics, chromatin dynamics. And it's a real pleasure to have him here. He started his graduate training at Osaka University, where he worked his way through his PhD. And then he moved to Geneva, Switzerland to do his postdoc with Ulrich Laemmle. Um, and after that, he was briefly a scientist at Riken Cellular Dynamics Lab before moving to the National Institute of Genetics in Japan in 2009 as a professor. And as I said at the beginning, he's moved his way up to the head of the Department of Chromosome Science. And I would just like to highlight the wealth of outstanding work that his lab has generated in the last 15 to 20 years. Over 100 publications, all of which seem to be of just astounding quality. And he is also very willing to collaborate. And you can see that from acknowledgement sections across probably hundreds of additional papers that he has shared many reagents and has been very generous with his time and with the work that his lab has done. So Kazu, please take it away. We're excited to hear about your research today.
Okay, so thank you so much. First of all, I'd like to thank the organizers for giving me this opportunity. Okay, so I'm very happy. I'm working in National Institute of Genetics in Mishima, Japan. So Mishima is small but nice city and quite close to Tokyo area. Just 50 minutes from Tokyo Station by Super Express train Shinkansen. And from our campus, we can see very beautiful Fuji Mountain here and NIG here. And NIG also very famous for cherry blossom sakura. We also have sakura book like this. Okay, so please visit NIG to meet our nice colleagues and also to see the beautiful mountain and the beautiful sakura. Okay, so our main interest, how is this long strand of genomic DNA organized in the cell and how genomic DNA behaves in living cells. So this is sort of classical textbook view, and DNA is wrapped around the core histone from nucleosome structure. And for a long time, the nucleosome had been assumed to be folded into this very regular 13-nanometer quantum fiber, solenoid or zigzag, whatever, and further helical folding. However, past 20 years, quite many evidences, including our Aquarium study and X-ray scattering analysis, have demonstrated chromatin is more irregular and variable structure and should be very dynamic. And now, more and more evidence to support this concept available from many labs. Okay, so chromatin is variable and dynamic structure. And recent study shows so chromatin is irregularly folded into condensed chromatin domain, especially in higher eukaryotic cells. So if we look at the euchromatin regions labeled by H3.3 halo, we see very condensed domain structure by super resolution image. So the question is how to form condensed chromatin domain in the cell. And to address this question, we focus on linker histone H1. So this guy. So globular domain with very long positively charged disordered region. So N-terminal and C-terminal. So C-terminal domain is longer, so 100 amino acid. So we focus on linker histone H1 because H1 can condense chromatin, especially in vitro. And H1 is the most abundant chromatin binding protein, and H1 is widely conserved among eukaryotes. So because of these three reasons, we focus on linker histone H1. Then question is, how can linker histone H1 condense chromatin in the cell? And if you open textbook, you will find this kind of figure. Linker histone H1 stably bind to the diet region of the nucleosomes, these crossing regions, okay? And then stabilize the 13-amino chromatin fiber. So this is a sort of textbook model, H1 binding model. However, so as you know, the 13-amino chromatin fiber is not the basic chromatin structure in the cells. And it's also well known H1 dynamically, not stably bind to the chromatin in living cells. Okay? So this was obtained by photobleaching experiment, flap experiment of linker histone H1. So we need a new model. And in my talk, we propose a new model. So H1 behaves as a liquid-like glue of chromatin. So the main contributor to this work is my PhD student, Masashi Mazoe-kun, with the help of Satoru Ide-san, Sachiko Taruma-san, and Kana Sato-san in my lab. Okay. So to study H1 behaviors in living cells, we used single H1 molecule image. And for this, endogenous H1.2 is tagged with hero tag and stably expressed in human RP1 cells like this. So localization of expressed H1.2 hero TMR is very similar to DAPI staining, suggesting the localization looks okay. So we picked up this H1.2 because H1.2 is the most major H1 subtype in RP1 cells. Okay. So there are mainly 8 subtypes, H1 subtypes in human cells, but this guy is the most expressed one in RP1 cells. And for single H1 molecule imaging, we sparsely labeled H1.2 hero tag with fluorescence ligand, TMR, such as TMR or other Janiria dyes. Okay, so we sparsely labeled histone H1.2 hero tag, and using oblique illumination microscopy or HIRO, which can illuminate very limited area in the cells with very low background. So we can see individual H1.2 molecules as very clear dots. I hope on Zoom still you can see their movement. Okay? So these dots show the single step photobleaching like this, suggesting each dot represents the single H1.2 molecule labeled by hero tag TMR. And the right one as individual nucleosome dots, okay, are labeled by H2B hero tag TMR. Histone H2B hero tag TMR, okay? And can you see the difference? So which one showed the larger motions? So maybe difficult. Let's look at individual dots, okay, single dots. So we determine the precise center of these dots by Gaussian fitting. and track their motion to make trajectories like this. And here is position determination accuracy is about 9 nanometers. Okay, so trajectory showed H1 moves more dynamically, and the nucleosome sort of looks fluctuating in around similar area. So we obtained many H1 trajectories from single cell. Okay, so H1 looks much more mobile than nucleosomes, and the nucleosome is more constrained, like this. And to quantify this, we calculated mean square displacement of histone H1. Okay, so MSD showed how the molecule move in a certain time. Then MSD plot showed on average H1 is much more mobile than nucleosome. Okay? So consistent with the previous reports done by photobleaching experiment of H1 in living cells. So flap experiment of H1 in living cells. Okay. Then we wanted to get more information from H1 trajectories. However, so H1 trajectories are too complicated to learn something. And to overcome this problem, we introduced machine learning-based technique, VBSPT. So variational based single particle tracking. So VBSPT can classify trajectory into several states based on hidden Markov model, and each state can be analyzed separately. Okay, that's the advantage of VBSPT. So we obtained H1 trajectories, and we see 3 states and classified by VBSPT, like this. So more constrained, the cyan one, and the green one is more diffusive, and the magenta looks like jump, dissociated, transiently dissociated from chromatin. Okay. So we see 3 states classified by VBSPT. And after classification, so we obtained a percentage of each state. And 30% of H1 we called bound because their motion is very similar to nucleosome, like this. And interestingly, 60% of H1 showed a linear increase in MSD. So MSD exponent is almost 1, 0.91. Okay? And we call this liquid like H1 because this linear increase in MSD is sort of characteristics of diffusive motion like liquid or fluid. Suggesting majority of H1 moves like a liquid. And the remaining 10% is called jump. So looks like transiently dissociated from chromatin. And we focus on this liquid like H1. The next question is, where does H1 diffuse like a liquid? So inside chromatin domains or outside the domain? So to answer this question, we need chromatin domain information in living cells. So in our previous study, so we visualized chromatin domains using in living cells using super resolution imaging PAR, photoactivated localization microscopy with histone H2B pH. Okay. Then we combined palm imaging of the nucleosome and single H1 imaging. Okay, so we labeled chromatin domains and H1.2 with different colors. So for chromatin domains, we used histone H2B PMJ. And for H1.2 motion, we used PAJF646. Okay. So, and using beam splitter system, we see nucleosome dots and H1.2 dots simultaneously side by side. And we determine the precise center of these dots by Gaussian fitting, and we map the nucleosome dots, and we track H1.2 motion to make trajectories. And we obtained more than 100,000 H1.2 trajectories from single cell. Okay. And we reconstructed chromatin domain by PALM, photoactivatable localization microscopy or H2B PMJ. And we also obtained H1.2 trajectories and classified by VBSPT. Bound cyan 1, liquid white green, and magenta jump. Okay. And this is reconstructed chromatin domains by PALM, and more white region is denser chromatin. Okay? And we put liquid like H1 trajectories on this reconstructed image, and you see beautiful overlapping. And the statistical analysis showed the denser chromatin domains have more liquid like H1 trajectories like this, suggesting chromatin domains are enriched with a liquid like H1. And this means H1 can move around within chromatin domains like a liquid. And then as a collaboration with Rosanne Colepard group at the University of Cambridge, UK, so we performed MD simulation of 12 nucleosome arrays and 12 H1.2. The cyan one is H1 molecules. Okay, so they are moving around within the nucleosome arrays. And from MD simulation, we found H1 interact with various regions of nucleosome. Not only that regions, but also linker DNA, nucleosome side, and back. And interestingly, so nucleosome diet is not the major H1 target in nucleosome. And we also found H1 interact with various regions of multiple nucleosomes simultaneously. Okay, so please look at this image. So single H1.2 molecule bridges 5 nucleosomes simultaneously. Okay. This distribution plot showed H1 interact with multiple nucleosomes from 2 to 8 nucleosomes simultaneously. So peak is 4 nucleosomes here. Okay, so H1 interact with various regions of multiple nucleosomes simultaneously. and suggesting H1 works as a glue of nucleosomes. Okay. Very long, positively charged disordered H1 region stick to the negatively charged nucleosomal DNA and works as a glue. And finally, so we wondered what happened to chromatin in the cells if H1.2 is depleted. And to address this question, we did rapid depletion of H1.2 using oxygen-inducible Degron system, which was invented by our energy colleagues Masato and Kanemax. And as you may know, AID system is really great system. AID tagged protein is rapidly degraded upon oxygen addition by proteasome pathway. And here, endogenous H1.2 is tagged with MAID and also mClover to monitor the depletion efficiency. And we also stably express histone H to be heterotag in the same cell line to observe the chromatin domains. And thanks to this AID system, 4 hours after oxygen addition, H1.2 disappears, and this was confirmed by Western blotting here. Okay. And after H1.2 depletion, we reconstructed chromatin domains by palm image. Okay. And rapid depletion partially decondensed chromatin domains. And to quantify this, so we applied this Ripley's L function or K function plot. And in this plot, if the dots are randomly distributed, no peaks here. But if the dots are clustered, show a peak like this. So we can quantify the distribution of dots or nucleosome. And rapid H1.2 depletion actually decrease this L function plot, suggesting less clustering of the nucleosome in depleted histone H1.2 depleted cells, consistent with our model. So H1 works as a glue. Okay, so let me summarize my talk using single H1 molecule imaging and machine learning based technique. We found 3 H1 behaviors in living cells. So 3 states of H1 behavior in living cells. Okay, the 30% of H1 relatively stably bind to nucleosomes. And this can be direct binding in the classical H1 model. And surprisingly, 60% of H1 dynamically bind to the nucleosomes and behave like a liquid within the chromatin domains. And the remaining 10% is jump. It looks like transiently dissociated from chromatin and back to liquid-like state soon. Okay. And MD simulation also suggests H1 bind to various regions of multiple nucleosomes simultaneously. Okay. And in my talk, just we focused on histone H1.2, but we also obtained very similar H1 behavior in using H1.20. So our results it's not H1 point specific one. And we also showed rapid division of H1 point partially decondensed chromatin domains. Yes. So, and putting all this together, we propose H1 works as a liquid-like glue of chromatin. Okay. So, very long positively charged to H1 disordered regions and stick to the negatively charged nucleosome DNA and works as a liquid-like glue. And our finding also suggests H1 glue can make chromatin compact and also accessible to proteins such as transcription factors. Yes, H1 glue can weakly make chromatin compact, and then so transcription factor can access even inside chromatin domains. That's our model. And our paper was published in Science Advances very recently. And if you are interested, please check it out for more details. Okay. And at last, I'd like to thank all my collaborators listed here. My PhD student, Masashi Masaiko, is a major contributor to this work. With the help of Satoru Ide-san, Sachiko Tamura-san, and Kana Sato-san in my lab. And the Rosa and Colepard group at the University of Cambridge performed really amazing simulation of 12 nucleosome arrays and 12 H1 molecules. Okay? And Masato Kanemaki-san at NIG kindly provided us their AID cell line, and Masaki Sasai-san at Kyoto University and SS Ashwin-san at Gitamu University, India, helped us H1 trajectory analysis. Okay, so thank you so much for your attention. I'd be happy to take any questions.
Awesome. That was really beautiful work. Thanks so much. I'll ask one question quickly. Do you think the C-terminal domain of H1 is sufficient for this liquid-like activity, or does it need the globular domain or the N-terminal domain to
Okay.
associate nucleosome?
Yeah, that's really exciting question. And at least what we know is if we truncate to amino acid substitution of C-terminal region, and then they can't bind to the chromatin. Okay, so we are sure that the C-terminal domain is very important for liquid-like behavior, but it's not so easy to separate the H1 binding and liquid-like behavior. That's only.
It'll be really exciting to see next,
Yeah.
like if you swap out different portions of the tail between the different H1s to try to determine why there are so many isoforms and are the differences between
exactly.
genes important. Yeah. We have Don and Aiden Olens raising their hand. I'm going to allow them to talk. If you'd like to unmute yourself, please ask your question.
Yes.
Okay. Hi, Kazu. Thank you very much for a beautiful talk. And can you hear? This is Don. Can you hear me?
Yes.
Good. Okay. And Ada is sitting right next to me. So I have first of all, that you've really established quite clearly the dynamics of histones in live nuclei. But let me ask a question that is very controversial, as you know. Do you believe that 30 nanometer chromatin fibers exist?
Yeah, as I said, so at least 30 nanometer chromatin fiber is not the basic chromatin structure in the cells. Of course, in certain regions, we can see zigzag pattern, zigzag fibers even in the cells. But our point is not the major or it's not the basic structure of chromatin. That's our opinion.
Okay,
And
thank
if you ask me the fiber exists in the cell, my answer is yes,
okay.
but not the basic structure.
Well, okay. We published a paper. First of all, you should know that we've shut our lab, so we're not doing any more experiments. Uh, we published a paper in Chromosoma in 2021 where we argue that the 30 nanometer chromatin fiber exists stably right adjacent to the nuclear envelope. And this was done by a cryo-EM where we rapidly froze cells. Now, we also related to this. We rapidly froze cells and then did cryo-EM with using in Wolfgang Baumeister's lab
With Peng Shu.
with Peng Shu, that's right. And it was very clear that we could see 30 nanometer fibers in these sandwich kind of structures that are next to the nuclear envelope called ELKs, or envelope limited chromatin sheets. And so the point is that we're making is, I mean, these structures look quite organized. And as I said, it was published in Chromosoma 2021. Unfortunately, we can't really show stuff, I guess. But if anyone wants to see it, they can see that paper and they'll see the data that we have. This is part of our larger interest, perhaps, on the nuclear envelope and how the nuclear envelope, particularly the internuclear membrane, stabilizes such structures as the 30 nanometer fiber adjacent to the internuclear membrane in these structures called ELKs. ELKs are, you can see them, we published quite a lot about them in retinoic acid treated promyelocytic cells, HL60 cells. And so we believe that there are regions that are very stable, and these are fibers that the cryo-EM showed that there's a sandwich in the ELKs of two layers of 30 nanometer chromatin fibers in a crisscross arrangement, two layers. Each layer is connected to a different internuclear membrane that has been sculpted in the nucleus. It's hard for me to describe without pictures. So I would say if anyone is interested, the thing to do is to look at our Chromosoma 2021 paper. So there are regions of...
I'm sorry. Thanks so much, but there are a couple other questions I want to get to before we get
thank you. That's really all I had to say. Thank you.
Thanks so much.
Thank you.
Last couple of questions in the Q&A from Marta. She asked whether the relative proportions of bound, liquid-like, and jumping H1, do those change across different stages of the cell cycle, or whether that distribution may be altered in disease?
Okay, that's a really great question. If we look at the heterochromatin region in mouse cells, big heterochromatin region in mouse cells, we see more bound fraction.
Okay.
So originally 30, and then in heterochromatin regions, then 40%. So we think this ratio can be changed from cell to cell type and cell state.
Excellent.
Okay. And in mitotic chromosomes,
Yeah.
When we look at H1 behavior in mitotic chromosome, no jump. So they
Okay.
are stably bound or liquid-like state. No jump.
Okay. Cool.
Okay.
Marta's second question was regarding H1's ability to interact with multiple nucleosomes simultaneously. Is there any evidence that that multivalent interaction changes under disease conditions or cellular conditions?
That's also interesting point. But we have none that. But definitely, we are very much eager to know that.
Yes. And I think really I'll just close here by saying, I mean, again, thank you, Shabi, thank you, Kazuhiro. This is beautiful work. I think you both highlighted proteins and complexes that are not as appreciated by the chromatin field as they should be. And absolutely, that's the case with histone H1, I believe. And I really look forward to the advances that you'll both be making in the future. So thank you once again for both getting up so early with us and coming to us from the other side of the globe. And in the case of me, coming to me from tomorrow, since it's already April 23rd where you are. Thanks again. And to the audience, thank you for attending, and we look forward to seeing you at the next Fragile Nucleosome. Thanks so much, everyone.
Great. Thank you, Jackson.
Bye.
Bye.
Bye.